Hello, 1, 2. Okay. Hello, everybody. Thank you for sticking around and for staying for a very exciting topic right before happy hour. So, I think LIMS. I was just discussing with the panel. I think LIMS. You're very excited when you don't have a system that is able to cater your needs, and you finally find a solution. After that, the good LIMS systems for me is something that is transparent and just enables you to do your job every day. We have four amazing people discussing the systems, the solutions, or the approach they followed, or they suggest we can follow to assess and find the right system for us. The first speaker will be Bob from Biopolymers facility from Harvard Medical School. Anoja will be discussing, and Bob will capture the process that he is following and his team is following to identify the right LIMS, but also I think what is key, to identify their needs, which I don't think is something very easy to put on paper. Anoja will be discussing the system that they have there at Stowers. Catherine as well from ETH Zurich, and Bill from Jackson will also have their own solution. We try to cover homebrew solutions, solutions that are also freely available, and solutions that are commercial. Looking forward to vivid discussion that will be in the end. All questions will be at the end, and I hope you enjoy. Thank you for joining. Bob.
Is this you? Jans, is this yours?
Oh, yeah.
Perfect. I might need that.
Oh, yeah, almost. Just make it full screen.
There we go. Sorry about that. So thank you, Giannis, for inviting me to be a part of this session. And I also want to thank all of our sponsors again for allowing the meeting to happen, and all of you for being here and giving us your time. So I'm going to talk mainly about the process of evaluating commercial LIMS systems and picking one for your laboratory, because that's what we're currently engaged in. Very briefly, a little bit of background. When I started directing our core, it was a long time ago. We were relatively small, offered 4 technology area services for about 40 labs and a few hundred scientists for $300K a year. Fast forward to the present, and we've grown quite a bit, offering a lot more technologies, a much larger user base, and significantly more samples and revenue associated with that. And so a big part of being able to accomplish that growth in a reasonable time frame was having a LIMS. So when I took over as core director, we had a very basic LIMS back then. It was my duty as the new core director to actually maintain the entire thing, so web server, database, et cetera. I'm not a programmer. I'm not a skilled computational biologist. So it became clear to me very quickly that that really wasn't a sustainable model for long-term support of the core. So I worked with the department and the school on hiring more qualified individuals to rebuild that LIMS from the ground up. And 22 years later, we're still using it. So why are we looking at replacing it? So one of the big ones is a single point of failure. So the entire system is basically maintained by one software engineer who knows the entire system, but No one else really does. So for business continuity moving forward, it's really critical to have a resource in place that can be supported by other individuals rapidly should something happen for that change. Other things related to that, obviously, to that person. Another big one is the possibility of new and improved features that we can have rolled out in a more timely manner than using a homegrown system. And then also the possibility of integrating with other HMS cores that would like to share the same resource. So before starting this process, some key questions that you can ask. Do you actually need a LIMS? I would argue that for almost every core now, yes, it's necessary in one form or another. And then, going further into that, is it because you need to scale for a larger number of users and samples, as we did in our case? Is your institutional oversight group interested in better risk management and oversight of your finances and who's using you? And then, of course, we all want to achieve maximum efficiency of the use of our employees in the lab, so we like them to do wet lab science and consult with other scientists to do good experiments and not be really bogged down with pushing a lot of paper or Excel sheets or other things that can happen if you don't have a really nice LIMS. And then also important, who's going to use it? Will it be just your core? Will it be all the cores? Will it be a subset of cores, as well as your user base? Excuse me. So financial support, both in the case of when you acquire it, as well as maintaining it, and then also doing the evaluation itself. For the evaluation project, will you, the core director, do it? Will someone within your institution be doing it? Or can you afford to hire a third-party contractor to do it? And in our case, that's what we did. So I'm sure the question is, how much does it cost? And I would say, depending on how complex your system is going to be and how many people it will serve, the evaluation probably will be more than 25K, but less than 100K. Other important questions: what is your timeline? So did you just get a big grant, you're setting up a new center fast, and you have to have something in place immediately? Or in our case, we have an existing system that's meeting our needs, so we can take our time and really do a thorough investigation, do this correctly, and hopefully come out with something on the other end that is a better solution. Building a coalition of support within your institution is really critical. So core staff, your super users, your biggest users of your core in terms of faculty and PI, if you have a steering committee, those individuals, and then other core directors as well. On the administrative side, most of us have a centralized department oversight for finance as well as the cores, so those individuals are key to bring on board. You may interface with your purchasing and accounts receivables, so it's important also to involve them. And then other individuals like risk management, audit services people, IT, for security and interfacing with your storage and high-performance compute infrastructure, and then other institutional leadership, which will vary a lot based on your institution, but some of those key players are shown here. So when all of this was done, this is a picture of our governance board for the evaluation project, broken up into all these different key areas of expertise and who we really need to have on board to succeed in doing the evaluation. It's a time-consuming process, and so that's why, if you can afford it, I highly recommend hiring a third-party contractor. In terms of the timeline, again, we originally thought we would be around 6 months. It's looking like it may go out as long as 18 months, but you really want to do it correctly. And again, that timeline will vary depending on how complex your workflows are, how many you have, et cetera. So there's a lot of steps in the process. I have them all here. I'm not going to go over all of them, but these slides will be available, I believe, for download through the ABRF website. So you're welcome to grab the presentation to look at them in more detail. The ones that I have underlined, I will talk a little bit about, because I think those are probably some of the key elements and most important. And then also important to note, we're not done. So we are at the step of actually beginning to conduct the in-depth demonstrations with our commercial vendor finalists. So I really won't be able to talk about the implementation and rollout process, which I'm sure everyone has lots of questions about, as do I, because we haven't done that yet. So first was to actually validate the business case of this, and I already covered a lot of these things. Why do we need the LIMS? What instruments are you supporting? How many samples are you pushing through your lab, and how many labs are you supporting? Defining all those benefits that I talked about previously, who's going to use it, and again, addressing costs, initial costs and ongoing maintenance costs. Another big step, and Giannis talked about this, was defining our requirements. So the third-party contractor came to us with an enormous Excel spreadsheet full of several hundred requirements. We immediately went through and deleted all the ones that were completely irrelevant to our operation, but we did leave some that we felt the vendors would ask us about, even though we will not have them. And so we prioritized them using this Moscow priority scoring process, where you define a requirement as must have, should have, could have, or will not have. So our final list had 290 requirements on it that we distributed to the different vendors. And then they were broken up into a number of different areas that I've noted here below, just general LIMS functions and ordering processes and so forth. You can see them there. We also had to map out all of our core workflows. So this is just one example of our post-library prep NGS QC and kind of what happens to a sample all the way through. So you can see there's a lot going on there. This was actually a really great exercise because these also helped us with organizing all of our SOPs and other documentation for the core. Harvard has an institutional SharePoint license, and so everything for the core is now really nicely organized and detailed there. So when I leave or others leave, it's very helpful in seeing how the core operates and all the different workflows associated with what the core does. We also had to map out infrastructure. So this diagram shows you all the different databases and the high-performance compute instances and various other things, accounts receivable and other institutional resources that we interact with. Next step was for the contractor to actually distribute the request for people to participate. So I didn't realize there were so many LIMS vendors nowadays, because I used to do a much more simplified exercise of this years ago and hadn't done it in a long time. But there was actually, that information was sent out to 36 vendors that they felt could potentially qualify for providing a solution for our core. We received responses back from 17 of those, and 15 of them wanted to participate. A few declined, and then there were some that, even after multiple outreach attempts by the contractor, they just did not respond to us. So that individual compiled all of the self-assessments that were done by the LIMS vendors that consisted of looking at all of those requirements and other information that he sent to them, compiled it into a scoring matrix, and this is just a tiny little snapshot of a piece of that. There was a lot more things that went into it, and then sort of generated this nice looking heat map, and we tried to identify who are going to be the top 4. And then we went out to our stakeholder groups and shared this information. So we had meetings compartmentalized by area of expertise, so we could focus on the parts that were relevant to those individuals in deciding on who the final 4 would be, and then they notified the companies of their status. So at this point, we're ready to conduct the really in-depth demos. So one of the things on a previous slide that I didn't mention was we built a lot of very detailed scripts that we want them to demonstrate for us. And I think that's really important because the contractor said in other scenarios, if you don't tell them exactly what you want to see, they're just going to show you their sales pitch, and you may or may not see what you would like. And then you go through a lot of back and forth and questions, and they're not prepared, and then you have to come back to other meetings. So that was one of the lengthiest processes that my lab underwent. Was, again, what do we want to see from them explicitly so that we catch every feature and every option and everything that we think we want to see in a LIMS. And that, again, was a really good exercise as well, because it pointed out some things in our existing LIMS that in the meantime we wanted to change while we're waiting to potentially replace it with another system. So this is the point where we're at in the process. These demos will occur over the next 1 to 2 months, and then we hope at the end of that we'll be able to make a decision, ideally to have more than one finalist so that we can actually do financial negotiations with regard to licensing fees and annual maintenance fees, et cetera. Hopefully at least one. And I will say, if the answer is none, that it wasn't really a wasted exercise, because we now have this incredibly detailed set of information that if we want to rebuild from the ground up a custom system that's cleaner and more efficient than the 2-decade-old system that we have all the necessary information to go to another contractor and present them with that and say, this is what we need, how long will it take, what will it cost, can you do it? So either way, I think it will be an improvement for the Corps. So acknowledgements, the funding to do this evaluation came from an internal award process at Harvard called the Foundry, so I want to thank them for providing those funds to the Corps. Our Associate Dean for Research, Carolyn Chamu, who happens to be here at the meeting, was a huge champion of helping us through to organize this project and get things going. Vincent Briere is our consultant from Verdience. John Gottleman, program coordinator, also has done a ton of work organizing meetings and scheduling things across this very diverse group of stakeholders. My core lab staff, who has, on top of their regular jobs, worked tirelessly to undertake this evaluation, especially my lab manager, Ashley. And then a lot of other cores, the HMS Single Cell Core, Nescent Transcriptomics Core, the HPC, and others that are all participating in this to help us pick a final solution. So, thank you.
I have like 5 presentations open. I had to open up the right one. All right, so my name is Anoja Pereira, and I'm from the Stowers Institute in Kansas City. We are a not-for-profit organization that does basic research. And we decided to go the custom route. We thought we were going to be brave and just do that. This speaker doesn't work. Okay. So a little bit about the Institute. We are a bit unique in the sense that we don't do just human and mouse work. We work with all kinds of weird and non-standard organisms. We got our first Illumina sequencer in 2007, and at the time there were just 2 vendors available to choose from as far as LIMS went. One was Geospisa and one was Genealogics. One did have the things we wanted it to do, and the other was like few $100,000 investment to start. Our institute had already had a dedicated module building group, so we were thinking, hey, we can do this. And at a past life, I also had some experience working with another LIMS team to develop a system, so we just took that challenge on. This is my fantastic team. We love working with each other, and we also have a lot of fun. We do all your typical NGS work, ChIP-seq, DNA-seq, bulk-seq. We do a lot of single-cell work. We do spatial work. We do multi-omics. We were basically an Illumina lab, but then very recently we added the Singular G4 and we are adding the Element AVT. If I can tell you two things to keep in mind if you go this custom route, is that a developer will build what you ask for. So you have to be very specific about what you're asking. As Bob mentioned, they spend a critical amount of time gathering all those requirements. So that's number one. The number two, you don't want too many cooks. You know, that was one of the hard things we learned. You have a whole group of people in a room. Everybody has different ideas. You have to have like point people. From the wet lab side, who should be making decisions? From the analysis side, who are the people, who's the key person saying, okay, this is how we're going to go ahead? That's very critical. What information should you capture? What are required fields that you want to capture opposed to like optional fields? And the workflow, you know, does it make sense for users, the wet lab staff, the analysts? And we also wanted to make sure that this system can last a while. So we wanted like every drop down to be configurable. We wanted to be able to add fields and change things as much as we wanted. We wanted reporting. We wanted emails to go out when we started making libraries. And of course, the analysts wanted API access. After gathering all the requirements, we also had our developers follow us around the lab because they had no idea what an Eppendorf tube was, you know. So they really appreciated that. All right, so the first thing, so the first thing the developers do is convert every ask that we had into this system from Atlassian called Jira. Every ask got converted to a ticket. And after every build, we met and started talking about it. Did it work? Is that what we wanted? And this was a long process. But it, again, like having the proper stakeholders in the room just to make sure everything was going well made a world of difference. The stage 3 was they have built the LIMS, and it was endless testing. There were so many bugs that we had to, you know, test and test and test to find out. But it was almost a light at the end of the tunnel because we actually had a system in place. And we had to make sure that it's capturing the information from the researcher side, from the wet lab side, from the analyst side, and we were having the proper permissions. The workflows are making sense. But the bug testing was painful, and it took a very long time. And at the end of it, though, we were pretty happy that we had a nice system in place. And we had multiple seminars to sort of introduce the system to the Institute. We put videos and documentation together so that people could follow through. And we went live with it. And even after going live, we were picking up bugs. So we were using Jira to capture those. So I'll just go through a few screenshots of what our LIMS look like. It's nothing fancy, but it does everything we want it to do. A researcher is able to log in and put all their chargeback information, their sample information. We have like a whole list of categories that they can fill out, but only they can skip if they want to and just give the required fields the core is requiring to do the processing. And then from the core view, a technician can log in and see what our queue looks like, you know, how many orders are in the requested stage, how many are ready for the person who's sequencing to run them. So this is like a very quick view of that. And when we are making the libraries, we can select an order. We can see all the information about that particular order. And then we can also, from the core side, put all the information that we want to capture about a sample that we are processing. This actually came into a lot of use. I don't know if you all remember, many years ago, Illumina had a recall for their RNA-seq kits. We had captured all that information in our LIMS, and we were able to get over $100,000 in credit because we were able to show that we had used that kit. And when running the sequences, we capture all the information there as well, and that way analysts can use that to process the downstream work. So some nice features that we incorporated. Like I said earlier, every drop-down is configurable. and analysts can put links, so everything gets saved, everything gets emailed out, researchers can log in and see where their data is at. Chargebacks, obviously. Every month we submit all these charges that gets auto-generated. These are all internal charges, so if you're paying any attention to that. And yeah, so finally, at the end of this process, we have this, we got a product that we are very proud of, and we use it every single day since like 2009. Researchers are able to log in, put their order requests in. The core gets all the information we need to process a given order, and the analysts can use the information that we provide to do the downstream processing, and the data gets available to researchers at the end. The chargebacks, reporting, search functions, all those things are available. This process took 2+ years to complete. So Bob's timeline of 18 months, you're pretty good. Will we do this again? Ask me at the happy hour. Yeah, so just to acknowledge, again, I absolutely love the team I work with. Wally is our head of computational biology, and Madeline was our point person from the analysis group to make the decisions. Jessica Witt runs the IT and research development team, and Alex Dean was our developer. Thank you.
How do you do that? Oops. My computer is sliding. Okay. Does it work? Works? No? Ah, yes. Good. Hi. So I'm Catherine. So I'm going to present to you about our system in Switzerland. So I'm from the ETH in Switzerland. Can you hear me? Can you see me? Yeah. So shameless plug. So tomorrow at 2:00 we have. I will present in the RG about. Last session there was a question about the duplicates, the sequencing, the different sequencers and how it affects the PCR duplicates. So we have a preliminary data with that about that. So 2:00 tomorrow. Shameless plug. Okay, so now I will talk about what I'm here to talk about. So our B fabric at the Functional Genomics Center in Zurich. I'm not calling it a LIMS because B fabric is not a LIMS, it's a way of life. So the Functional Genomics Center, and one more thing. So thank you very much also for being here today. It's very exciting, this topic of LIMS. And I'm sure Chris will be faster to make mammoths with a very properly working LIMS system. Okay, so the Functional Genomics Center, it's a core facility of two universities, the ETH and the University of Zurich, and we are actually a multi-omics facility. So we are offering genomics, transcriptomics, proteomics, metabolomics, genome informatics, and proteome informatics, and data integration. And then, so we have around 200 projects every year, new projects every year, and genomics orders, we have around 1,200, staff is 50, 8,000 registered users, and the different subgroups. So we just moved to a new building, so we're quite proud of it. It's very nice, especially in the snow. So our genomics portfolio, so we have all the Illumina toys, as everyone else here. and also some sample prep robots, and this is our proteomics portfolio. So I'm sure you're all here today because you woke up one day and because you purchased a new sequencer, and then you just woke up and like, oops, our Excel sheets doesn't work anymore. I cannot pull correctly. That didn't happen to us. Why? Because we're Swiss. You know, the land of efficiency and clocks and organization and stuff like that. So when the center was founded, my boss, who is here today, already conceptualized also having the B fabric that will support the whole thing. So we call it B fabric, or the Swiss Army knife for life sciences. So I will explain to you a little bit more. So it's conceptualized to be fair data compliant, which is findability, accessibility, interoperability, reusability. So the data is there to be that you can find it again and use it again. And it's conceptualized for multi-omics facilities. We have implemented it since 2004, but imagine that the FAIR paper was just in 2016, so we were far ahead. One functionality is real-time, on-the-fly lab notebook functionalities. Also, storage and annotation of all data produced at FGCSet. And it also offers a structured way of data retrieval for the user and access controlled through a web portal. And also, it allows you to dynamically couple external applications with the system. That's why it's very, so that's why we can still use it after 20 years or so. So why is implementing LIMS in a core very challenging? It's because when you're working with a system, it can really help you work, but it can also control how you work. And the problem is, the feature is core facilities, we really need this flexibility because we're not like companies who are just turning out, PCR, whole genome sequencing libraries and sequencing it. So we have a lot of different ifs and buts and, little small sample, low input. So we have to be able to be flexible and actually. So the LIMS should be able to accommodate new workflows and applications, but since we're super flexible, not all of our workflows can be SOP'd. So when you think about it, a badly designed LIMS can become a problem and not a solution because suddenly you cannot work. You cannot work anymore just because you cannot document it. You cannot use it, so you say, I cannot do it. Like, accounting and SAP. So it's always like accounting, like, okay, I cannot do that because I cannot put it in SAP, like that. So we don't want to do that, right? Because we're flexible, we're a core facility. Yeah, so these are just the complexity of the orders and the samples at FGCSet. I also think about LIMS as a bag, because I think I like bags. So I think about it as a bag, and it actually holds all your data, right? And you have to hold everything, organize everything, and you should be able to go with it, somewhere. So, for example, these are all the things that the requirements of that all the things we have to take care of, like sample submission, project management, order submission, purchasing, instrument reservation, sample tracking, data analysis, communication, tracking, internal communication, on-the-fly communication, billing and invoicing. It's a lot. I mean, it's a lot of things to put in the bag, right? And you have to have a nice bag. And then also there is a challenge. So the picture above is the genomics workflow, and the picture above and below is the proteomics workflow. And you can just imagine that we even think differently because we present ourselves differently. How do you put it how do you converge these two worlds in a LIMS, in a project management system? Yeah. Well, so this is the scheme of B-Fabric, of how B-Fabric works. But I'm not going to talk about that because that's not my area of expertise. I didn't build the system, so actually my area of expertise is actually complaining effectively about the system, effective criticism that actually what I need gets implemented. And that's really the hardest part because the data scientists, they're data scientists. They have no clue about what we do in the lab. As they said, they have not even seen an Eppendorf tube, right? So, I mean, we had years of discussion about, is it called sample or extracts? So I mean, it's years and years and years of discussions about that, or about Excel uploads. Do we do Excel uploads? And they will always say, no, we can't do Excel uploads. It's too complicated. Anyway, so so apparently there's a certain way to talk to them, to talk to our data integration people. You don't say Excel upload. You say, can you please have a table in the B-Fabric and cut and paste from Excel? That works. Yeah. So there are certain things that we need to translate. There's a translation, like a Rosetta Stone thing, because it's really two different worlds. So I'll just show you quickly how it. So some screenshots about of the system. So because it really reflects how we work. So our basic unit is a project. So we're very ambitious because before we don't just get orders, we have projects. And then within so basically they put who they are and who will pay and so on, and what is their research question, and then we discuss their research question, and then after that we talk. We can then decide on which technology they need to answer their research question, and that's the project. So within the project, they can submit orders, and the orders can be proteomics orders, metabolomics orders, or whatever. Not whatever, but genomics or NGS or something else, or PacBio, whatever they need. And then within this order is a bit more specific because this is basically whatever it is here, that's what we will do. But usually we review it to before we run an S4 flow cell because we only have an NovaSeq 6,000, not yet an X. So we try to say, are you sure that's what you want? And then we put an offer just to make sure that they really understand that this is expensive. And then they will agree to it, so they accept it, and then and then they can deliver the samples. And then after and then basically after the order, then they will register the samples, they annotate the samples. And then, and then we will review the order. That's what I'm saying. There are billing information here. You might charge them, so I put it top secret. And all this time, we have a milestone thing going on. So basically, every time we click on something, they will receive an email where we are at the step, which step we are. And then, for example, we document if, so when we receive a sample, we QC it first. And then if the sample fails QC, we say, it fails QC, we'll put it on hold. And then we will communicate with them through B-Fabric as well. And then everything is documented. And then we have an order summary for the lab. And for this, we can see all the states of every order. And then we put all the orders together that can be processed together and make a plate. And then after the library prep is done, so and the library prep, for example, the QC, we have also beamer scripts that beams the data from the fragment analyzer or the tape station. And then from there, we know how to pool or normalize and stuff like that. So everything is there. And then for the sequencing, we have then the run manager. And so we can put everything in, so for example, we can pull the samples with the right barcodes in the one run. And then when after it's run, we have the run metrics, and all the run metrics from the Illumina machine is also stored there. So we know what, how the run looked like 3 years ago. And then, so this is a feature again. We have a seamless forwarding of the data to the bioinformatics team who sits with us as well. So when we are done, when the data is out, they know, and then they can start analyzing it as well. And then here, we also know what kind of bioinformatics analysis has been done on the order. And this is tracked and documented, and then it goes to billing and invoicing, and then so we configured it as well for Spatial. So for example, here the ROIs we could connect to the sequencing run, and then for single cell we also have a new. An add-on basically that we know how many cells we loaded and the cell ranger runs and profiles. We have that too. And so this is, and then we also have, what is a very important feature of bFabric is the communication thing. The communication, the ability to communicate internally and externally. So basically we gossip there, no, but very professional of course. Because some of our users can be our colleagues later. So, so some of them also have difficult samples. And then if you're like, fuck, this is shit, it's, yeah, it's true, it happened. So when we hired the person, we actually had to check what we wrote in his project because, to be safe. So we have to be, yeah. But we document everything. So and then we also the external communication, the external comments are also there, and this is forwarded to everyone involved in the project. So the PIs and so basically, for example, that a single cell run, it's very bad. No viable cells. And then basically they said, but please go ahead because that's all I have. And then so we will put that in as a comment to the documentation, but it will also go to their PIs who will pay, and then everyone can see that this is the responsibility of the person because we said this is against our advice, but you went anyway. So what I mean is it also helps a lot in conflict management. Because we document all the what we think, and then there is no discussion because everything is there. It's cleared as well. So some calculations. Well, it's not cheap. So we have been running it for 20 years, and with 40 full-time employees. So we have spent approximately 6 million francs or dollars, $6 million. And but we calculate that we have saved 9 million. We have our value. The value that we get is 9 million francs because of efficiency. So this means we already earned 3 million on it. Yeah. So that's the last slide. So the major challenges for me, so summarize in developing and implementing a homebrew LIMS is actually, so the first thing is the communication between the biologist and data scientist. Yeah. But I'm still very, very proud when our data integration group leader can discuss with me single-cell sequencing with feature barcoding. I'm proud. And so core facilities, we need flexibility. LIMS can streamline the work, but it can also constrain it, so we have to be careful. SOPing workflows are really hard. training of users and employees. I mean, this thing there's so much functionality with bFabric. Even I don't know all. I don't even know how to use it, the whole thing. But well, so you actually need a PhD to run it. Yeah. So it's also difficult to prioritize what features you actually need because there are so many things that can be nice, but then it takes time to implement it, and then at the end, it's also costly to implement new features. And also to determine what information you really need captured. And so another very important challenge is that it's very difficult to retain the people or to the developer retention because basically it's like the people. So it's very hard to hire people who will work on it because we're competing with banks or Google and stuff like that, and we can't pay so much because we're academic. Yeah. So so this is our team and Ralph and Shan, who's actually the how you say the one who developed bFabric, who's not here today. Thank you. Sorry. Oops.
Come on, computer. Come on, computer. Cool. Alrighty, well, I'm going to tell you a different kind of story. My name is Bill Flynn. I'm from the Jackson Laboratory. I think I'm now envious of the LIMS systems we just saw presented. So I'm going to tell you a little bit of a story where we are about maybe 2 years past where Bob is. But so just to give you a little bit more context as to what is JAX, because I think people know that we make mice and sell them to people. So we have two major campuses, one in Bar Harbor, Maine, one in Farmington, Connecticut. But that's actually just a little bit of where most of our research is done. We actually have campuses in Japan, in California, and in China, and we serve not just our own research investigators here at JAX. We also work with University of Maine, Northeastern, Tufts, University of Connecticut. We have about 75 faculty, and all of this is supported by about 16 core services, and these are probably the major ones. Some of these have sub-core services. You can imagine at JAX, something like reproduction and preservation does a lot. And so there's many different core services in there, same with Center for Biometric Analysis. But so I'm in the single-cell biology lab here, and I'll tell you a little bit more about what we do in a second. But the interesting thing about our 16 cores is that they are supported by 27 LIMS and electronic notebook solutions. And so JAX just celebrated its 94th birthday. And so you can imagine there's a few reasons why we might have a lot of LIMS. One is just colony management is hard, and we need to keep track of lots of mice and cages and things like that. And that's one of the reasons we have one LIMS. But a lot of the other reasons are because, faculty came and no one told them what to do, so they brought whatever they were doing before. And is it even a LIMS that they brought? Maybe it's just an electronic notebook that they're paying some tens of thousands of dollars every year for and didn't tell anyone. Also because more recently some grants, right, are now mandating that you have kind of a larger institutional support for some kind of system. But really the major two reasons is just because we had a lack of a centralized mandate. And especially as JAX was getting really started, in really productionizing mouse cells, right? There was just an absolute lack of solutions. So we had a lot of homebrew things inside. But so what does my core do? We do essentially, I would say, the three major ways I think you can profile cells or characterize cells, right? You could do it in a droplet, in its tissue where it came from, or in a dish. And so we do try to do pretty much everything we can. So that's unbiased, spatial, unbiased single cell and spatial technologies, targeted spatial technologies. And then high throughput functional characterization with some automation and microscopes and stuff. And so because of this complexity, we were essentially one of the core targeted labs to essentially guinea pig out a solution. And so our institution essentially gave 3 labs, one that was fairly low complexity, something like cell engineering, right? Essentially, you have a cell line, you'll start with, you'll make a CRISPR knockout, and essentially it's just validate the CRISPR knockout, expand and freeze. That's fairly simple, fairly linear. Something that's fairly complex but low throughput, something like mass spec, where you can do a lot of different things, different mass spec workflows, but unfortunately at JAX at least, mass spec is a fairly low throughput service. And then our service, which is all of these things with quite a lot. We have about $4 or $5 million in revenue in our service every year. And so after a 4-year ad hoc search through a partnership between our kind of core oversight committee and IT that focuses around research, we essentially searched for the one solution to rule them all, and it has its pluses and minuses. And ultimately, that was at L7 Informatics. And so there's essentially 2 ways that you can go about interacting with L7. One way is you pay them lots of money and tell them what you want, and they will do it for you and then give you a solution. The other way is that they will give you the software and then give you some training, and then you figure it out. And so the low complexity team worked with L7 directly. That worked somewhat. It took a long time. We chose to be the guinea pig of guinea pigs and chose to work with our internal development team, which started out with 0 people, with 0 expertise, and now, after about a year and a half, is a team of 4 core developers, a project manager, and 2 full-time contractors. And so we can actually now ramp up that full-time contractor to kind of manage our load. And so it's actually been a pretty good transition recently, but it's been a long ramp up. And so what is L7? So this is their L7 is the company. Their product is the Enterprise Science Platform called ESP. And so it does a few things. It does workflow management, and it's all based on a variety of little apps that kind of all are modular and work together. It also does inventory and kind of location management, like what room is your sequencer in, things like that. It also can do pretty much anything that you could do in Python or R or Julia or anything like that. And it can also easily integrate with other systems through APIs. But its main thing is really auditability and chain of custody tracking. And so the nice thing, pretty much what they sell you is it's flexible, it's extensible. They don't subscribe you to a particular data model, so it's essentially BYO data model. And it's fairly useful at modeling complexity, and it's very computational friendly. You can run things on the cloud, you can run things on your local HPC, you can run stuff locally, dashboards, things like that, custom applets. It's also important, though, to know what isn't L7. And so L7 is doesn't do billing and finance tracking. So we still use iLab, and we interact with iLab through an API. It doesn't really do project management, right? It does like sample tracking, but it doesn't really do much project management. And so we still use Confluence and some Jira and email and other things. And it also doesn't really do time management or scheduling, yet at least. And so until they do that, I would love for someone to tell me what you guys do, because this is a huge thorn in my paw. And so here's kind of an example of what L7 looks like. I don't really want to get into the details of all the things it can do, because it's quite a lot. But the major whole thing about it is creating workflows and linking those together into what they call workflow chains. And workflows themselves are kind of a concatenation of different protocols. And each protocol is essentially just a glorified Excel sheet. So it can be essentially rows by columns, where each row is some entity that you care about, but it can also track things like stuff about that spreadsheet, for example, so metadata about the spreadsheet. And then each workflow can link together in very nonlinear ways. And so all of these kind of orange arrows that you might see linking these green boxes together are essentially ways to transition from one workflow to another. And that could be in something like single-cell RNA-seq, right? You start with a cell suspension, maybe you'll end up with gems that you put in the freezer for 4 days. Then eventually you'll get cDNA and libraries. All of those will happen at those transition points. And so that works well in theory, but we had some transient challenges. And so the first one, like I said, right, is we were the guinea pigs of the guinea pigs. And so it took a long time. It took about, in all honesty, it took over 12 months for us to get a team fully trained and to really get their feet wet with the system. It's now substantially much more agile and productive. I would say more agile and productive than working with L7. And so in the short term and the long term, we're actually saving a lot of money going this way. That said, these people need to essentially be full stack developers. They need to know Python, they need to know JavaScript, they need to know SQL, things like that. Additionally, the one thing that we didn't really anticipate it was how much of an upfront core commitment like there was going to be. And especially in the absence at the beginning where it was just about a 0-person team, we didn't really have an interface between the wet lab folks and the developers, and we definitely didn't speak the same language. And so I think that's a theme that you guys have heard quite a lot. They definitely didn't know what an Eppendorf tube was. Honestly, when I started 6 years ago, I didn't know what an Eppendorf tube was. But luckily, I think we have mostly solved that issue now that the team is up, and we've had some stumbles. That said, there are still some remaining challenges, and these are mostly, I would say, kind of on us, a little bit on L7. They do claim that it is complete flexibility. But in practice, there are some concepts that are fixed and that you need to adhere to. And the major ones are really about what is the notion of a project and how does that relate to an experiment, and some other things like what is a customer, things like that. Additionally, I was talking about transitions between these different workflows. That is where all of the secret sauce happens, and you need to kind of rethink the way that your wet lab protocol goes or your bioinformatic protocol goes. Because anytime you want to transition from one type of entity, a suspension to a cDNA to a library, that all has to happen at those junctions. And so sometimes in real life, there's some additional pieces of information you want to capture as you've transitioned cDNA to fragmented library that you've amplified. You actually need to fudge where that actual boundary is in software to make it really easy to actually manage those transitions. But once you kind of learn that, that's fine. The bigger challenge, which we haven't really accomplished yet, or overcome yet, is our own worst enemy is ourselves, because the more complex, you can make it as complex as you want. It's bring your own data model. So that means you can think of any complicated situation you want, and you can implement it, and it will work, maybe. But then you have to deal with that working solution that's terrible. And so the more you try to abstract things away, the harder it is to really deeply track information. So here's kind of one example. We started 18 months ago. We're like, yeah, 3' gene expression, all the bells and whistles, high throughput, cell surface antibodies, multiplexing. We can model it like this. This didn't work, and it spent like 2 months trying to make it work, and it just didn't work. And so now we have something that looks like this to manage about 12 different protocols that we can group into maybe 3 big structures. But doing this process has kind of given us hope, has given us direction. So even though it's been a little bit of a stumbling block, we've learned quite a lot, and now we are kind of putting things into gear. So we anticipate by the end of this year, we'll be fully up and running with all of our dissociative methods and Visium, and then things like high content screening, codecs, that kind of stuff will come next. So that's it. This is my team. These are the main 3 people that worked in addition to my team on the IT side. That's Dave and Janet and Alan. And thank you. That's it.
I wanted to thank our speakers and you for sticking till the end. If we have a little bit of time for a couple of short questions. Yeah, there's someone here.
Anoja, and if there's an equivalent, Catherine, have you been able to get your system approved for GMP or CLIA, or is there an interest?
At Stowers, we don't, because we don't work with human samples.
That's my only question.
Okay.
Are there any other questions? My question is, after all this effort, if you were to start today knowing what you know, which approach would you have followed? Would you do exactly what you did, or, and this is for anybody on the panel to pick it up?
Can I ask a clarifying question? Are any of the homebrew solutions open source or available?
I asked as well.
Okay, because depending on that answer, I would maybe change my answer.
Yes.
Okay.
Mine is not, and I asked the institute about it. They said they don't want to do any kind of support. I don't want to spend time answering any questions.
Yeah, I jumped on the B-Fabric queue.
Okay.
I think we will also not, I mean, they can, Ralph, you have to help me.
Yeah, it's available. It's available at no cost for non-profits, at cost for profits. Okay, so it's kind of it's a defined license, so it's not.
So there is a license for also non-commercial and commercial use. So there's different types of licensing. The reason is partially what has been mentioned, because there has been sort of a control in terms of support efforts. But the idea is clearly that there's also interfaces, APIs, so that people can also attach their own solutions. So it's by no means a closed system, and there should be a community at one point to help develop and broaden it, because we clearly started with Omics solutions in mind, but we all know that the workflows are getting more interdisciplinary over time, and we have to attach also different types of data and input.
Thank you. If there are any other questions, thank you all for staying, and I would like to thank our speakers once more. Have a wonderful evening.